is what happens in the S phase during G1. What is happening is the regular processes which of the cell, which is to produce proteins. Okay, so we're inside the cell, inside the nucleus. The DNA is just strung out. It's not condensed into its chromosomes, and transcription is going to be for one gene. Okay, so I think humans have around 20 or 30,000 genes, and transcription is for one gene, because one gene is going to equal one protein. Okay, so one protein is the result of this whole transcription translation process. Okay, so transcription. We have one gene of DNA. Okay, from beginning to end. We're going to have markers on those genes. Transcription factors are what they called. So we have RNA polymerase, we have the Trna box, and we have a whole bunch of different proteins that all assemble at that promoter and region there. We have RNA polymerase, which I already said that one. We also have the enhancer, which is a little further down. And it's going to come into this region of the Trna box. This piece of DNA is going to bend around. Okay, so if it was to move up here, so it's going to look like this. And that enhancer is going to touch back on itself. And when that happens, when this enhancer, the same one that was right there, connects here, that allows a chemical reaction to take place. It's going to allow RNA polymerase, which is a lot like DNA polymerase, to start copying this one gene. Okay, and so this one gene might be for, eye color. It might be for, hair color. It might be, the many different genes that we have in our body. It's going to copy this one piece of DNA. Okay, so what does RNA polymerase do? So, similar to what DNA polymerase does, we have two strands of DNA here, well, one strand, two-sided, come in, and what happens is there is a little place that is going to separate out the DNA, and in that amount of time it's going to build new DNA or new nucleotides. Okay, and so what we get is we start getting this long chain of single-stranded RNA. So RNA makes RNA polymerase. Okay, and then instead of leaving it open, the RNA polymerase actually closes the DNA back up and returns it to its normal state. So what we have is just this long piece of RNA that is produced, and when the RNA polymerase gets to the end of the gene, it'll stop, and this piece of RNA will then leave. And it heads over to what is called editing. So editing is a very important step in transcription, and transcription is going to be finished once that RNA is produced. So we're going to have this long strip of RNA, and editing is going to take place, and it's going to cut out the exons and the introns. So exons are going to exit, and that means that they are going to be, whoops, I didn't mean to erase that, the ones that are going to head out and be part of translation. So if this is an exon, this is an intron, this is an exon, and these are just regions of DNA. Now the introns are going to stay inside of the nucleus. And these are going to go back up, and right here where these new nucleotides are being found, right here, these little guys are coming in at a very fast rate. And so there might be A's and T's, like this might be an A, this might be a U, C, a G, okay, because RNA, there's no T's. And those come from the introns when it's cut out. So what we're left with is just the exons, so they're all exons. It's going to shorten this, and this is going to be called our mRNA, which the M stands for messenger, and it's going to exit the nucleus. Okay, so after it exits the nucleus, we are going to meet up with the second type of RNA. So our first type of RNA is our mRNA. It's going to leave the nucleus. Our second type of RNA is rRNA, and that is ribosomal RNA. And so it is actually nucleotides that go through and form these two subunits. And our RNA is a ribosome. And this is where proteins are made. And then our last thing that is part of it is the tRNA. And tRNA kind of are made up of DNA just the same way. They're going to have three little nucleotides that we're going to call the anticodon right here. So this might be an A and a C and a G. Okay, and this is our anticodon. The mRNA that lands right here, each three of these are going to be called our codon, and they're going to be attracted to each other. So what happens is the tRNA comes in and it binds with the codon if it's the right code, so it has to be an A matching with a U, a C matching with a G, and a G matching with a C, and a U matching with an A. So mRNA, our nucleotides are A's with U's and C's with G's. And this is really important because what it's carrying is a amino acid. And all of these have a different, there's 23 amino acids, all of them have a different one. And as these things come in and join, these tRNAs come in, they're going to start building this polypeptide chain of amino acids that eventually is going to fold into a protein. So our three types of RNA are going to be mRNA, rRNA, and tRNA. Protein is made at the ribosome through anticodon and codon matching up, and the amino acids making a polypeptide that turns into a protein. These proteins could be used to make your heart beat. They could be made to make the skin color the way it's supposed to be. Whatever that gene, that one gene, was being coded for, is going to be produced in that one protein. Okay? Now we have some different mutations that can occur. Okay, and so there are mutations